Theivanai Ammal College for Women. Learn, live, and lead.
Welcome you all to the molecular biology class. This is Sridevi, Department of Biochemistry, Theivanai Ammal College for Women, Villupuram. In today's class, we are going to see the central dogma of molecular biology. What is central dogma of molecular biology? It's nothing but it is defined as the flow of genetic information from DNA to RNA, RNA to protein. It's called as a central dogma of molecular biology. So here, let us start with the DNA. DNA is nothing but deoxyribonucleic acid. So this DNA is going to get the information which is going to produce the protein molecule through transcription and translation. So there are two steps involved to convert the DNA into a protein molecule. The first step is transcription, where the DNA, that is the deoxyribonucleic acid, is transcribed into a ribonucleic acid. Here, if you take this DNA is produced through the replication process. What is replication? It's nothing but copying of parental DNA molecule to form an identical DNA molecule. And this DNA is made up of 4 letters. It's nothing but the 4 bases comprises of adenine, guanine, thymine and cytosine. Okay. So this DNA is going to carry this information and it is converted into a RNA, that is nothing but the ribonucleic acid, which is made up of again the 4 bases or 4 alphabets. Here adenine, cytosine and guanine, whereas the thymine in the DNA molecule is replaced with the uracil. Okay. Next, this RNA is going to be translated into a protein molecule in the form of polypeptide chain. So if you compare this RNA and the protein molecule, here this RNA is made up of ribonucleotide sequences, whereas the protein molecule is made up of 20 amino acids. And here, why it is called as a translation? Just like that of the normal English language, here translation, if you want to translate something from the Tamil or to the English or English to the Hindi, like that, there what happens, the language translation will be taking place. Similarly, here if you take the RNA molecule, the language encoded in the RNA is the ribonucleotides, whereas the language encoded in the protein molecule is nothing but the amino acids. So the nucleotide sequences are going to be translated in the form of amino acid sequences to produce the polypeptide chain to form a protein molecule, which is the final product in the central dogma of molecular biology. Okay, so here this central dogma of molecular biology shows a single flow of information, as already pointed out, from DNA to RNA and RNA to the protein molecule. So based on this, there are 3 different forms of central dogma molecular biology was identified. One is called as unidirectional flow of genetic information, circular flow of genetic information, and the last one is the reverse flow of genetic information. Let us see one by one. What is unidirectional flow of information? As we discussed earlier, here if the genetic information flows from DNA to mRNA, then mRNA to protein molecule in a single flow, that is called as a unidirectional flow of genetic information. Who identified this? That is a DNA. More double-stranded helical structure of the DNA was identified by the scientists Watson and Crick. The same Watson and Crick identified this unidirectional flow of genetic information in the year 1958. And what happens in case of the circular flow of genetic information? Here, according to this, reverse to this the Crick model. Okay, here this was identified by the scientist Commoner in the year 1968. Here, what happens actually? The DNA has to be converted into RNA, and RNA has to be converted into a protein. That is the final product in the central dogma of molecular biology. Here, what happens? This protein molecule again converted converted into a RNA and RNA converted back into a DNA. So that's why it is called as a circular flow of genetic information. Let us see what happens in case of reverse flow of genetic information. So this reverse flow of genetic information was identified by the scientists Deming and Baltimore in the year 1970. So they identified this reverse flow of genetic information in the RSV, that is a virus called as a Rose sarcoma virus. Since it was identified by the Deming, this also called as a Timinism. So here what happens, here the double stranded, we know the RNA in certain cases it also present in the form of double stranded structure. So that double stranded structure of the RNA molecule converted into a DNA, and this DNA is converted into a RNA and RNA into a protein molecule. So here actually the DNA has to be converted into RNA and RNA into a protein. Here what happens, the RNA acts as a genetic material. Usually the DNA, we will be having a two. One is the DNA, which is nothing but, as already said, deoxyribonucleic acid. Another one is the ribonucleic acid. So this RNA is called as a genetic as well as a non-genetic RNA. When the RNA called as a genetic RNA, so if the RNA is going to act as a precursor or the substrate or the source for the synthesis of the protein molecule directly without the involvement of the DNA molecule, then it is called as a genetic RNA. Non-genetic RNA is nothing but it is going to act as an intermediate in transforming the information, that is the genetic information. See, if you take this, see here, we are having the DNA molecule. This DNA is converted into RNA. It may be mRNA or it may be the tRNA or it may be the rRNA. So this RNA, if it is converted into a protein molecule, here the RNA is not directly involved in the synthesis of the protein molecule. Here it takes the help of the DNA to produce the protein molecule, which have a unique property and unique role in the inside the cell to carry out the structural as well as the functional properties to lead the normal life. So in this case, the RNA, this RNA is called as a non-genetic RNA. That is the difference between genetic and non-genetic RNA. And coming to this is the difference between the unidirectional circular flow and reverse flow of genetic information. I said there are 3 steps involved in the, that is, first one is the replication, second one is the transcription, and third one is the translation. So replication is nothing but copying of the parental DNA to form an identical daughter DNA. Whereas the transcription, here what happens? Deoxyribonucleic, 2 strands of the DNA is going to involved. So involved in the transcription process, but not both the strands will be acting as a template strand. So what is template strand here? Template is nothing but the strand which is going to produce a complementary RNA strand. That strand is called as a template strand of the DNA. So out of these 2 strands in the DNA molecule, one strand is going to act as a template, which is called as a nonsense strand, whereas another strand is going to act as a non-template strand, which is a sense strand. So this DNA, after converting into a single RNA molecule, single strand RNA molecule, this RNA will be acting as a template for the synthesis of the protein molecule through the translation process. So here we will be getting this RNA in the form of mRNA, which is called as a messenger RNA. So this messenger RNA carries a message from the DNA and it will be transferred by the tRNA. So that's why it is called as a transfer RNA, and where this protein synthesis, final product of the central dogma of molecular biology, takes place means inside the ribosomes. Whereas this transcription or the formation of the RNA molecule takes place in the nucleus. So once it is produced inside the nucleus, it comes out of the cytoplasm and takes part in the protein synthesis inside the ribosomes of the rough endoplasmic reticulum. Here, this RNA will be having the sequences, nucleotide sequences, ribonucleotide sequences. These ribonucleotide sequences, which is present in the form of base sequences, 3 letters or three bases joined together, and it represents a one codon. Okay, this codon is going to tell what is the amino acid that is going to be transferred by the tRNA. So I said that the ribosome plays a very important role, or ribosome is going to act as a protein synthetic machine. So these ribosomes, we are having two subunits in the ribosomes, larger subunit and the smaller subunit. In the larger subunit, there will be three different sites will be present. One is called A site, another one is a P site, and third one is called as a E site. What is this A, P and E? A site is called as the amino acyl site, whereas the P site is called as a peptidyl site, whereas the third one, E site, is called as the exit site. So what is the role of these three sites during this translation process inside the ribosome? Means this A site only, the incoming amino acids will be added. And what is the role of this P site? Is a site where the peptide bond formation takes place. Why we need this peptide bond? Because we are going to produce a protein molecule, and this protein molecule is made up of a series of amino acids. All these amino acids are linked and joined together with the help of a bond called as a peptide bond. That peptide bond formation between the two amino acids takes place in the P site. Once the peptide bond formation completed between the two amino acids and the tRNA, once the role is over, it will be moved out of the ribosomes with the help of the exit site. So these are the three sites which is involved in the protein synthesis, which is also present in the ribosomes. Okay, so after this, what happens after? That is decoding the information from the DNA molecule through the RNA. Finally, it will be converted into a protein in the form of a polypeptide chain. This is called as a central dogma of molecular biology. Thank you.